Okay, good morning. And we're going to go ahead and get started now. We've got most everybody here. And we're going to continue our discussion on the structures of the cell. And hopefully we'll see, make it a little more interesting than it has been. Of course, we won't be having, hopefully, any power failures or interruptions of the internet in between here. So, okay, you're still having trouble with the lockdown browser. Well, okay, let me take a look at your test and see what we can do about that. So let me mark that. Okay, I can, if you're having trouble with lockdown browser, just shoot me an email, let me know. I've already seen here. Okay, let me see what I can do, and I will fix it so that you can take your exam. So don't panic, don't worry about, I'm sure you want to take your exam, but I can go ahead and see what I can do to fix that lockdown browser problem. So I don't know why it's doing what it's doing. Never really.
Is this Dr. Sprode, is this a timed exam? Because mine is saying like unlimited still.
Oh, it's supposed to take an hour. It's supposed to be an hour with I think 10 or 15 minutes of extra time allowed there. These exams shouldn't take more than 45 minutes to do. The average person does it in about 45 minutes.
Sure.
So for 50 questions an hour plus 15 is more than adequate.
Perfect.
So just don't look at the timer. Some people look at the timer and go, and you go, and then start worrying. But if you just go ahead and work through it, it's okay. So, okay, that answer your question? Good. Okay, let's get started then. Let me bring up the PowerPoints I want to look at first. We are in cells. Okay. And let me get back out of here. And okay, everybody should see the slide. It says diffusion limits. And the reason we're talking about that today is that there are three terms here that we were involved with. When we're talking about water movement in a cell, we're talking about hypertonic, isotonic, and hypotonic. Hypertonic solutions occur when the solute concentration is greater than what's in the cell. So like if you had, if you took a cell and dropped it into a 10% salt solution, which is 10 times greater than what the cell has, the cell has like 0.9% sodium chloride, and the outside of the cell, the interstitial fluid should be 0.9% also. If you take a red blood cell and drop it into 10% salt, a 10% salt solution, well, you know what water does. Water always goes from the high water side to the low water side. So there's more water inside the cell than there is out, and water will go out of the cell and it'll shrivel up, it'll crenate. The thing about the human body is that we gain and lose water all the time, but we generally don't gain or lose a whole lot of salt. And so if a person becomes dehydrated, they're going to lose water, but they're not going to lose the salt. Now, when we become dehydrated, our salt concentration goes up because there's less water. Consider if you have a beaker full of salt water. If you have a beaker with a 10% salt solution, and you let it evaporate. When it evaporates halfway, if you have a liter of water with 10% salt in there, if you evaporate it down halfway to 500 mils, your salt solution is now 20% because you cut the water in half. And the more that evaporates, the saltier it's going to be until you end up with nothing but dried salt in an empty beaker. And our body is sort of like that. The water concentration outside the cells can fluctuate. It can go up, it can go down. If the water concentration drops, the salt concentration goes up, not because we gain salt, but because there's less water. And so when we get a saltier solution, cells will always give up water to the saltier side. So a hypertonic solution, we don't want to give somebody salt water to drink because it draws water out of the cells. The cells are going to shrivel up. Now they can recover. What we like is for the water inside the cell to match the water outside the cell. As long as the salt concentration in the interstitial fluid, which came from the plasma, is the same as the salt concentration inside the cell, everything's good. If there's water movement, it's going to be, no, you're not going to see any net change. A third condition, a third term is called hypotonic. In a hypotonic solution, the outside of the cell has a greater water concentration than the inside. You know, a person that decides to, for whatever reason, chug a case of water in one bottle after another. What's going to happen here is you're going to flood the plasma, because where's that water going? It's going into the plasma. Everything we eat and drink goes into plasma. So that water goes into our plasma, leaks out into the interstitial spaces, and causes greater water to occur outside the cell. What does osmosis say happens when you have high water? You go from high water to low water. And since there's less water inside the cell is going to expand. And if you have someone who is overhydrated, someone who can't pee, or they're taking in too much fluid, for whatever reason, that water ends up in the cells, and the cells are going to start absorbing that water and expand. And given sufficient water, those cells can explode. Red blood cells, if placed in distilled water, will explode because they'll take in so much water, they'll lyse, they'll pop. We don't want that either. And we've demonstrated that in the past in lab, where we take red blood cells and put them on a slide, and then put a drop of distilled water on the slide with them, and all of a sudden the cells, which were visible, now turn into nothing but a red smear because they've all popped. This is why we don't give patients distilled water in their IVs, because that would create a hypotonic environment. It would lower the salt concentration in our plasma. So the three terms here: hyper, too much solute; isotonic, the solute levels are balanced; and hypotonic, too much water. So this is what, a little better explanation of tonicity. Isotonic solutions have the same osmolarity as the inside of the cell. The solute particles are the same number of solute particles inside and outside, so there's no pressure to move the water in either direction. Hypertonic solutions have greater solute particles outside the cell, so water is driven out of the cell to try and reach equilibrium. And in the hypotonic solution, there are fewer solute particles outside the cell, and so water is going to go into the cell. And of course, then we have lysing, rupturing of the cell. When the cell shrivels up, it's crenation. That's the membrane stays intact, but the cell is going to get much, much smaller. So you have to be very careful in treating patients with the right kind of IV solution. Normally, the normal IV line, just to keep the line open, is what we call normal saline, and it has the same salt concentration as plasma. It's an isotonic solution. We use it as a blood expander. we would give a hypertonic solution to someone, one that is saltier than normal, if they are swollen, if they are suffering from edema, if they have like congestive heart failure, for example, or they've been stung by a bee, or they've eaten something that they're having a body-wide allergic reaction to, you may consider giving them a hypertonic solution of saltier saline, because it makes sense, to draw water out of the cells and back into the blood. But the one you don't ever give to your patient is a hypotonic solution, because that will cause the red cells to explode. And we don't like that. So these are all types of passive transport. Remember that osmosis is passive transport. Osmosis is a category of diffusion. Diffusion is passive. Facilitated diffusion is passive. Osmosis, the movement of water from high water to low water, is also passive. It's also a subset of diffusion. But there are occasions where simple diffusion isn't good enough. There are occasions where we want to move something across the membrane in the opposite direction of diffusion. Diffusion goes from a high concentration to a low concentration. We all know that. We've seen that demonstrated on the PowerPoints. But what happens when you want to go from the low concentration against the gradient to the high side? What happens when you want to go uphill? And so this involves what we call active transport. Now, active transport occurs, there's three categories that cause it. The substance that you want to get across the membrane is too big to fit into any kind of channel, too large, or it's not fat soluble, or more than likely, in most cases, you can't move it passively against the concentration gradient. If you have lots of sodiums outside of a cell, that's a technical term, lots of sodiums outside a cell and a few sodiums on the inside of the cell, and you want to get rid of those sodiums that are inside the cell, they're not going to diffuse out on their own. The concentration gradient says, hey, let's all go into the cell. Those sodiums inside the cell don't belong there. So the cell has to actively pump them out into the interstitial spaces, or there's a great deal more sodium there waiting for them. You have to move against the concentration gradient with active transport. Active transport requires ATP to physically move these substances. Now, active transport covers, there's the subset of that is known as vesicular transport, where we actually use vesicles to bring something into the cell or out of the cell, take something out of the cell. Both of those require ATP. But when I say active transport, when I say active transport, I'm talking about the physical pumping across the membrane. Vesicles are different. We'll see in a second. So active transport, we actually have pumps, carrier proteins in our cell membranes that act as a pump. It will pump one direction or it will pump both directions. We have what's known as an antiport, where we go in, we pump one thing in while we're pumping something out. So we have, like a two-lane highway, one going one way, one going the other. Symports are both going in the same direction, but they're different substances too. Like, for example, and you'll see this later on, sodium and glucose, totally different substances. One's an ion, one's not. One's a salt ion and one's a sugar, but they're both going in the same direction. So we have antiports and symports, but they're always going against their concentration gradient. For example, cells like to have lots of sodium outside and lots of potassium inside. There's always more potassium inside a cell than there is outside. But potassium will leak out of the cell through diffusion. But we don't want it to stay out there. We want to get it back. So we have to pump it back into the cell. We're still going against the concentration gradient for potassium. So, but when we have a pump that will pump the potassiums back into the cell from the interstitial fluid. And these little pumps are embedded in the membranes of our cells. Their role is to pump against the concentration gradient. They go from low concentration to high concentration, and they have to use ATP. Cells use about 40% of their overall energy budget for ATP. Okay, I'll get back to that one in a minute. Now, cells use about 40% of their overall energy budget to pump. Now, there's two types of transport. There's primary active transport, where we're actually using the ATP to power up a pump. The pump is plugged in, if you will, in the membrane, and it's powered up, and it's busy pumping sodiums one way or potassiums another way or glucose or something else. That is primary active transport. It gets its energy directly from the ATP. Now, secondary active transport is a little bit different. Secondary active transport comes from maintaining a concentration. Now, what does that mean? Well, when you pump sodium, when sodiums leak into a cell, and cells don't want sodium inside, just take that as a fact right now. Most of our cells, well, actually all of our cells, are surrounded by sodium, lots and lots of sodium on the outside of the cells, and they all have potassium on the inside. Sodium and potassium are the key ions that show up in our cells. Sodium's on the outside, potassium's on the inside. The cells don't want sodium inside at all. They don't want sodium inside the cell. They don't want potassium outside the cell. So when sodium leaks in, we have to pump it out. When potassium leaks out, we have to pump it back in. When we pump the sodium out, it will leak back in through diffusion. But when it does, it creates a symport to drag along glucose. And this is what secondary active transport is all about. By keeping a constant, if the cell had its way, all the sodium on the outside would leak into the cell and we'd reach equilibrium and the cell would be useless. But the cell is busy pumping. When the sodium leaks in, we pump it back out. And it's going to leak in again because we're keeping a high concentration on the outside. You're pumping the sodium out, and all it's going to do is leak back in. You pump it out, it leaks back in. You pump it out, it leaks back in. But when it leaks back in, it will drag along glucose. And so we're getting a benefit by pumping the sodiums outside the cell. We keep the concentration, we maintain the high concentration of sodium outside. And as it does leak back in through simple diffusion, it's going to drag along glucose. And if we didn't keep that concentration high, then we couldn't drag glucose in with it. And that is secondary active transport. Secondary active transport doesn't require any more ATP. We use the ATP to pump the sodiums out. But as they leak back in, they bring along glucose. Now, primary active transport involves special carrier proteins that act as pumps. They physically change their shape in the presence of ATP to force ions across the membrane. We have pumps for calcium. We have proton pumps. In fact, we find proton pumps in our stomach all the time. Proton pumps are how we maintain stomach acid in our stomach. The protons, remember, a proton is a hydrogen ion. The strength of an acid is determined by the number of hydrogen ions it release, and these proton pumps are pumping out these hydrogen ions in our stomach through what we call gastric pits all the time, keeping our stomach fairly acidic on the inside, pH of 0 to 2, somewhere in that range. If a person has acid reflux, one of the medications that they take, and I've said this before, maybe what's known as a proton pump inhibitor. And Protonics is a proton pump inhibitor. I think Prevacid or Pepcid, one of those may have a proton pump inhibitor in the mix of it. The proton pump inhibitor shuts down this pump. It shuts down the actual pumping of hydrogen ions across the membrane of the cell. So we have calcium pumps, hydrogen pumps for the protons, and the most well-known pump is the sodium-potassium pump. This is the #1 pump that our cells deal with, the sodium-potassium pump. And as long as we have energy, we can pump sodiums out of a cell and keep potassiums in. This is very important to us. The sodium-potassium pump maintains the sodium concentration on the outside of the cell. See, our cells are all charged. We have positive charges on the outside of our cell and negative charges on the inside. It allows us to do things. Those electrical charges, we are electrically driven. And the sodium, as long as we keep high concentrations of sodium outside and high concentrations of potassium inside, we're going to keep this positive and negative balance. If our cells were allowed to reach equilibrium, they would cease to function. If the charges became balanced, they wouldn't work. So, sodium potassium pump, we know more about the sodium potassium pump. Make sure I didn't miss anything on that slide back here. Yeah, there's a sodium potassium pump in place. There's another one. What does the sodium potassium pump do? It pumps sodiums out and then it brings potassiums back in, but it requires ATP. And we need the pump to work for our muscles to be able to contract and our nerves to generate and send nerve impulses. We need the sodium, we need the potassium. We got to keep high levels of sodium outside and high levels of potassium inside. If we reach equilibrium, the cell can't function. So here's the pump. Now there are some important factoids here you need to remember. I don't know why, you just do. For testing purposes, you need to remember this. I've never had a situation where anybody's come up to me and asked me about how many sodiums get pumped out each time from the pump, but the answer is 3. The sodium potassium pump is an enzyme embedded in our cell membranes. Lots of these enzymes are embedded in the membrane, and it will always pump 3 sodiums out and 2 potassiums in. You need to remember that. 3 sodiums out and 2 potassiums in. See, because sodium's always leaking into the cell through diffusion. Sodium always finds a way in. There are sodium channels that will let in the sodium again and again and again. And potassium's always leaking out. There are potassium channels that will allow the potassium to leak out. We don't want that. So we're always pumping sodiums out and bringing potassiums back in. Now the pump will change its shape when energy is applied to it. Our normal pump looks like this. It has an opening for 3 sodiums. The little yellow things are sodium. It will hold 3 sodiums that will go into the pump. And then when ATP is applied to the pump, it will release those sodiums to the outside of the cell. So there's our cell, there's our pump. there goes 3 sodiums, and we use energy. You know, it's open in that shape. The ATP allows it to open to the outside of the cell. The sodiums go out. When it opens up to the outside, potassium can come in. The pump has receptor sites on the inside of it for sodium and potassium. We don't do it at the same time. These are going to be it's going to be like an antiport arrangement. One goes one way, one goes the other way, but not at the same time. Sodiums go out, the gate opens up, the pump opens up to the outside, potassiums come in. And then we keep opening and shutting the pump on either inside the membrane or outside the membrane. So we never open up a hole into the membrane, but we're always letting sodiums go out and potassiums come in. It's always 3 sodiums out and potassiums in. They're moving in opposite directions because we have to maintain high concentrations of sodium outside the cell and high concentrations of potassium inside the cell. It's absolutely essential for our cells to function. So it's not so much that you got to, for testing purposes, you got to know the numbers 3 and 2. But it's more important to understand that we're always moving sodiums out and potassiums in. And then when we get the sodiums out, the sodiums leak in, and then we pump them out, and then we can use them to piggyback with glucose, and we can get our glucose in that way. And so here we see it. Here's one that's not, moving so rapidly. There's our pump. There's the slot for the 3 sodiums right there. When the pump when it opens up to the inside of the cell membrane, inside the cell, the sodiums go in. And when it opens to the outside, sodiums leave. Potassiums come in. Then the pump opens up to the cytoplasm, and the potassiums come in. But that doesn't happen unless we have ATP. Once we get those extra sodiums out in the interstitial spaces, it will diffuse back across the membrane, but it will do so in what we call a symport. The symport has a special notch on it, or a location, a receptor site for sodium and glucose together. It won't let sodium in by itself. It won't let glucose in by itself. But when they're both present, this sodium glucose symport, will let both sodium and glucose come into the cell. And so this is one of the reasons why we continue to pump the sodiums outside. It's just sort of a side benefit of the sodiums being outside. See now, glucose is a large molecule. It's too big to diffuse across the membrane on its own. But if we have a specialized structure that will carry it over, like this symport is a protein embedded in the membrane, and it's huge, relatively speaking. It's bigger than the glucose molecule. The glucose molecule will land, when the symport is open, glucose can land on it, and then the symport will close to the outside and open to the inside, and glucose can come in. Yeah, glucose is a big molecule, but it will. This is its workaround. It goes through this specialized channel protein, and that's how we can get around the fact that glucose alone can't just slide through the membrane. So this is how we get glucose in the cell. Is this what insulin does? Does insulin drive the production of these symports to bring sodium in and glucose at the same time? It may be exactly how it's done. This may be the type of carrier protein that insulin tells the cells to make. And when our blood sugar levels get elevated, we ate something, we ate carbohydrates or something for breakfast, and we release insulin from our pancreas, and the insulin says, okay, this is how we get that sugar in the cell. We're going to make more of these symports. We're going to bring in some of that sodium that we've got out there, and it's going to piggyback with the glucose. So we also may just be making special channel proteins just for glucose. Not quite certain how this is all happening, but it's important that we use, for whatever reason, sodium and glucose tend to piggyback a lot. As any other way, glucose, you're absolutely right. Glucose is a big molecule. It's not going to slide across that membrane. It's just too big. Plus, glucose is water soluble. Water soluble substances can't get through the lipid layer of the membrane. There's no way. It's going to be rejected. The only way it can get through is to have this special carrier protein in place for it. Okay. Good question. Now, the other approach here is that involves the use of ATP is the use of vesicles. These are bubbles. These are bubbles that form within the cell that are, and the walls of the bubble are made of the same structure as the cell membrane. So it's the phospholipid bilayer. And we can move things into the cell or out of the cell. And we form this bubble called a vesicle, made up of the same phospholipid bilayer, and it will fuse with the cell membrane. So you're never punching a hole in the cell membrane, but you're still bringing stuff in. So things that are really, really large molecules, cell fragments, debris, because when the cell dies and breaks apart, all of its friends and neighbors go eat it. So, they destroy the fragments. If a structure, if an organelle wears out and breaks down, it's destroyed that way. If we encounter bacteria, we can engulf the bacteria and destroy the bacteria through the use of these vesicles. Now, there's two types. Endocytosis, where we're moving stuff into the cell. Think of an amoeba, engulfing something, food, or a white cell. Now, what we see up here at the top, we see this structure here. This white cell that just came up here is eating these bacteria, and it takes it inside and destroys it. So it comes along, engulfs it, brings it inside, destroys it. And what it does is it extends, a white blood cell will extend its membrane around the bacteria, the virus, the fungus, the protozoan, the dead or dying cell will extend its own membrane around that and form a bubble inside, and then start breaking it down. We have what's known as phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is food eating, pinocytosis is water drinking, and receptor-mediated is a specialized receptor just for certain types of molecules. Exocytosis is how we move stuff out of a cell. We form a vesicle, like we see down here. Here comes the bubble. It fuses with the membrane. It forms part of the cell membrane with its structure and opens its contents to the outside. We never lose the integrity of the cell membrane. So this is endocytosis. We can go up and we can act like an amoeba, and we can engulf something, food, something solid, a fragment of a cell, a fragment of an organelle, maybe some other food item. We can engulf that by sending our the wall. We can bring the sides of the cell membrane up and around it and form a vesicle, a bubble. And then once that vesicle's inside, then we can attack it with our own digestive enzymes. If we're going to, the same thing applies to liquids. If we are going after a particular liquid, we can engulf it and pinch off the vesicle right here like this, or we can have a specialized receptor site just for certain types of items, certain types of molecules, that only those will become taken in. Now, exocytosis is a little different. With exocytosis, we've already created a vesicle on the inside of the cell. Perhaps we've made some proteins, and we're bringing those to the surface of the cell. Now, we don't want to punch a hole in the cell, so this vesicle comes up and fuses with the cell membrane and then opens up. But you'll see that even though it opens, it has not broken, it has maintained the structure of the membrane itself. It fuses with the membrane, opens one side, this is still closed, and whatever's here can go out into the extracellular fluid, around the interstitial fluid around the cell. So if we've made a protein, we want to release that protein, we simply bring it up in a vesicle to the surface of the cell, fuse with the membrane, release the contents, and there it goes. And this is what it looks like. This is at 100,000 magnifications. We see a protein, for example, being released from a vesicle that has fused with the cell membrane. The cell membrane briefly extends now all the way around the inside of the vesicle and back up the other side, but we're still not open to the outside. So active transport and some sort of movement with a vesicle, either exocytosis going out of the cell or endocytosis going into the cell. And within the cell, so we've seen the membrane. This is all about membrane up to this point. Inside the cell is the cytoplasm. Cytoplasm, as I said, is often called the cytosol. It is the cell sap inside the, it really is sort of like a syrupy mixture inside the cell. It's water and salts and proteins and sugars and all this other stuff thrown together in this sort of goopy little mixture here. The cell sap, the gel, when we say cytoplasm, we're sort of think of something like hotcake syrup or something like that consistency. We have all sorts of structures inside the cytoplasm. We have glycogen, which is the polysaccharide version of glucose. We have vacuoles. We have salt crystals. We also have organelles. Organelles are like little organs. They're inside the cell in the cytoplasm, and they do things for the cell. They release energy from glucose. They string together amino acids to make proteins. They release those proteins. They contain, the organelles contain digestive enzymes to break down foreign substances or digest food. So these organelles are located, scattered throughout the cytoplasm. Now the organelles are broken out as, do they have a membrane or are they membrane free? Most of our organelles have membranes. The mitochondria, which you may have heard of, what we call the endoplasmic reticulum, or ER, because saying endoplasmic reticulum over and over again is really annoying. The Golgi apparatus, what are known as the peroxisomes and the lysosomes. We also have the ribosomes, the cytoskeleton, and something called the centrioles. We'll talk about these here. This is a mitochondria. This is where glucose is ultimately oxidized to release electrons. because that's where energy comes from in the cell. We break down sugar, we break down glucose, we oxidize it, we strip off we release energy by stripping, by breaking covalent bonds. And so all that potential energy the valence electrons have is released, and that happens at inside the mitochondria of a cell. We take the energy from the glucose and convert it to energy that we can use, usually in the form of ATP. So we call them energy conversion factories, or the term that everybody likes to use, well, most everybody's. The power plant of the cell, well, it's not really a power plant, it's taking energy from one form and converting it to another form. The mitochondria have a two-layer membrane, and all the work is done on the inner membrane, and the mitochondria produce the bulk of the ATP for the cell. Because remember, ATP is what it's about. ATP is where the energy from glucose goes. We park the energy obtained from glucose in ATP. We transport the ATP around our bodies, and we use that ATP to apply the energy to things we want to do, like the sodium potassium pump uses ATP for energy. Now remember, we'll get more into this in chapter 24, but we only got 7 seconds worth of energy from an ATP molecule. We can't store it, so we're always making more and more all the time. I guess that's redundant, but we're constantly making it. How's that sound? Okay. And this is what the mitochondria look like, and it is this area right along in the inner membrane where most of this ATP production takes place. Another organelle, this one doesn't have a membrane, this is the ribosome. The ribosome is made up of protein and a type of RNA cleverly called ribosomal RNA. And most of these ribosomes are attached to the endoplasmic reticulum. The ribosome is where we make proteins. That is the site of protein synthesis in our cells, and that's in the ribosome. Now, the endoplasmic reticulum, where most of our ribosomes are found, are a series of tubes that surround the nucleus. While it only looks like it's covering, one part of it, it totally surrounds the nucleus all the way around, like a ball. So it completely covers the nucleus. And the endoplasmic reticulum, these tubes, are either embedded with ribosomes, where we end up calling them rough ER, because you have all these little ribosomes sticking on the surface. That's what those little dots are, where it says rough ER. Or we have smooth ER, where we have the tubes, but no ribosomes. They have two totally different functions for us in our cells. The rough ER is where we make our proteins. This is the site of protein synthesis. The proteins are assembled in the ribosome, and the ribosomes are on the rough ER. And so we put together our proteins and our phospholipids in here, and then we move them to where they need to be. The smooth ER is different. We use it, among other things, we store a lot of calcium in there. It turns out that we need lots of calcium for muscle contractions. We have to have a great deal of calcium for muscle contractions, and we store it in the muscle cells in the smooth ER. We also make our cholesterol-based hormones in smooth ER. And the steroid-based hormones, all derived from cholesterol, are made in the smooth ER. We transport fats in our smooth ER. And a very important role of smoothie R is detoxifying, breaking down chemicals that we encounter. Now, all of our food and all of our water, and everything we eat, everything we drink is absorbed in the small intestine, not in the stomach, but in the small intestine. And as it's absorbed, it goes immediately to the liver. The liver cells have a very large percentage of smooth ER to detoxify, to break down the chemicals that show up in our food and the things we drink. I mean, consider this, consider medications. Let's say you have a headache. So you take 2 Tylenols or Tylenol, knockoffs. So the average dose for an adult is 1,000 milligrams of Tylenol, 2 500 milligram tablets or whatever. Now, that goes into our stomach and into the small intestine. It's absorbed, and the liver gets a hold of it. Now, the liver will detoxify. The liver will break down some of that Tylenol. Doesn't break it all down, but it breaks down some of it. Liver doesn't like Tylenol to begin with. The liver will break down some of it, or Advil, or Bayer aspirin, or Aleve, or anything like that. Any kind of medication. Let's just say it's Tylenol. What comes out of the liver and goes back into the blood supply, what remains of the Tylenol is what's going to be useful for our bodies as a pain blocker. That's what Tylenol does. It blocks pain. So the average adult dose for Tylenol is 1,000 milligrams. Whatever comes out of the liver is what's left over to work in our bodies. We don't get a full 1,000 milligram dose. We get whatever the liver lets us have. Now, an adult liver can process an adult dose. But consider a child's liver. The reason that children have different medication doses than adults, is because a child's liver is smaller. It can't process as much of the medication. So plus the body stature is smaller, so they don't need 1,000 milligrams. We take 1,000 milligrams of Tylenol because of our adult stature. We have to be able to have that go throughout our bodies. It doesn't know where to go. Pain medication doesn't know where to go. Inflammatory medication doesn't know where to go. It just goes, and wherever it encounters inflammation, it's going to work on it. Or wherever there's pain, it's going to work on it, but it's a systemic approach. Now, in a child, you would give a smaller dose, smaller body stature, smaller liver, because a child's liver can't process 1,000 milligrams of Tylenol. It'd be too much. It would be an overdose. It would have a significantly detrimental effect on the child's organs and on their liver. You would never give 1,000 milligrams to a child unless you didn't like them. So you would never give 1,000 milligrams. The same thing with an elderly patient. Their dose is based on their medication doses are frequently based on what their liver can detoxify. The liver is a detoxifier. It cleans up things for us. Alcohol, as a consumable. If people drink alcohol, where's that alcohol going to go? It's going to go to the liver. It goes to the liver first. The liver will process the alcohol and break it down. It will ultimately break the alcohol down to carbon dioxide and water, and that's fine. If the average healthy adult, if they ingest 1 ounce of alcohol, it will take the liver an hour to clean that up and break it down. Okay, so that's how much alcohol you have in 1 beer, 1 glass of wine, or 1 mixed drink. An hour. It takes it an hour to do that. If you increase the pace of consumption, 2 ounces, 3 ounces, the liver can't keep up. And so a person who drinks more than 1 ounce an hour of alcohol is going to, there will be a bit of a price to pay. Since the liver can't keep up with the amount of alcohol that's coming in, it will get backed up. As it breaks down the alcohol, the byproducts it's breaking down that normally are as fast as they're made, they're broken down. Things like formaldehyde, embalming fluid, if you will. As fast as that's one of the byproducts of breaking down alcohol. It's an intermediate step, but it breaks down as fast as it's made, as long as it's the rate of alcohol coming in isn't too great. But if you drink 2 ounces, 3 ounces, 4 ounces, you're going to see an accumulation of these byproducts. And the more alcohol you ingest over a short period, reasonably short period of time, the consequences the next day are what we call a hangover. That's your liver getting caught up with all the chemicals that it was making as it's breaking down the alcohol. So, if your patient wants to know why they have a hangover after they've had too much to drink, it's because their liver can't process that fast. So, so whether and again, whether it's a glass of beer, whether it's a 12-ounce bottle of beer, or it's a mixed drink, or if it's a glass of wine, an ounce of alcohol is still an ounce of alcohol. The liver can only do an ounce an hour. So anyway, so we break down chemicals, we break down food additives in here, preservatives in our food, coloring agents in our food are all broken down in our liver. And it's all done in the smooth endoplasmic reticulum of our liver. We also break down glycogen. Our liver does a whole lot of things for us. The liver is going to be where we have glycogen stored, and we break down that glycogen into glucose. So we see the smooth ER does a lot of work for us in here, not just in the liver, but in other structures within the cells too. So anyway, that's the smooth ER. Now the Golgi apparatus is adjacent to the rough endoplasmic reticulum. Now the Golgi apparatus, when proteins get made in the rough ER, they're produced according to the directions from the DNA, and each ribosome is making a protein, a different protein at a different time, and they're all lumped together in vesicles coming off the rough ER. Those vesicles go to the Golgi apparatus and they get sorted out. So while you have a vesicle containing 24 different types of proteins going into the Golgi, what comes out at the other end of the Golgi is all the same type in a vesicle. It's sort of like FedEx at one of their ports, like over in Memphis. Memphis was their original sorting facility. Planes pull in from all over the east and all at Memphis, and all their packages are sorted. And a plane will come in with packages from 3 states, and then they'll be sorted. So everything going to LA or Seattle or Chicago or Phoenix or wherever are then sorted out and going to different planes. So a plane going to Seattle will be taking packages just for the Seattle area, or a plane going to Chicago will be taking packages to Chicago and not something for Louisiana. We sort it out. That's what the Golgi does for us. Every vesicle coming off the Golgi is going to contain the same types of proteins: hair color, eye color, skin color. Are they, the same type of enzyme, enzyme A, enzyme B, are they amylase, hydroxylase, carboxylase, whatever? Are they enzymes? Are they the lysosomes? They're all going to be the same. And when they get to the Golgi and the Golgi releases them, there's 3 ways they'll go. They'll either leave the cell, stay inside the cell, or stay in the membrane of the cell. So This is what the Golgi looks like. This is what it looks like for real. It's a series of tubes that run back and forth like this, and they filter out and collect all of the same type of protein. Because there's our vesicle from the ER with a all mishmash of proteins. And here, every one of these vesicles contains exactly the same protein. We can either release them or to the outside. We can contain them inside the cell as the lysosomes and digestive enzymes, or we can embed them in the cell membrane for other use. And this sort of walks you through it. There's the rough ER. There is a vesicle filled with proteins, and they get sorted here. And this shows you the 3 different pathways. You can release them through exocytosis. You can incorporate them into the cell membrane, or you can move them over here to become a lysosome. Lysosomes are structures that contain digestive enzymes that break things down. And speaking of breaking things down, here we have something called the peroxisomes. This is something that uses brings hydrogen peroxide into play. And you're all familiar with hydrogen peroxide. You go down to Walgreens and you can look at, you see it there in the brown bottle on the shelf. It's like 88 cents a bottle or something like that, or at Walmart. It's a brown plastic squared off bottle. And we pour it on cuts and it foams and it doesn't hurt like alcohol does. And it helps to foam out the crud in there and it kills bacteria. And that's hydrogen peroxide. Something similar happens here with peroxisomes. Peroxisomes are used to break down what we call free radicals. A free radical is a molecule that picks up a charge where it shouldn't. Maybe it's a negative sodium. Shouldn't be a negative sodium ion. It should be always a positive. Perhaps there's a charge on glucose. Perhaps another molecule has broken apart and formed positive and negative ends where it shouldn't. Those are free radicals. They shouldn't have formed. And because they're charged, they're highly reactive with other structures. And these free radicals, if they roam through cells, can cause a great deal of damage to the cells and to the other organelles inside the cell, may be linking to disease or malignant stages here. So we want to get rid of these free radicals. We detoxify the free radicals, and we use oxidase and we use catalase to do this. Oxidase, the enzyme oxidase, is going to convert these free radicals into hydrogen peroxide in our cells. Hydrogen peroxide, however, inside our cells is dangerous. Hydrogen peroxide only works well for us on the surface of our skin, and then only in very small, in the concentration that you would buy at the store is only like 3% hydrogen peroxide. It still bubbles and foams and does its job. But you wouldn't drink it, you wouldn't inject it. Every so often you'll see a scam that comes along of hydrogen peroxide, the secret energy booster, because it has an extra oxygen molecule. H2O2, hydrogen peroxide. It's got an extra oxygen on it, it's like water, but only with an extra oxygen. So the scam argument is that, well, extra oxygen means more energy. And there have been lots of scams where people take hydrogen peroxide tablets. I don't know how they work, or they have it injected directly into their plasma. Again, not gonna work. It's like going to one of these. You see these topical applications for your muscles that contain ATP, but ATP applied to your skin doesn't do anything. You know, if you're rubbing it on your skin, it's not, at ATP, first of all, it's not going to retain any energy. It doesn't work. Our bodies don't work that way. Hydrogen peroxide, as soon in the case of a peroxisome, as soon as we detoxify a substance in the hydrogen peroxide, in the peroxide, we have to break it down immediately. And we use a second enzyme called catalase, which breaks it down to water. So the peroxisomes are our free radical destroyers, and we like that. And when it works, we end up releasing, converting the toxic substance, whatever it is, into water. We also use peroxisomes to make and break fatty acids. So, because fatty acids, where do we get our fatty acids for our triglycerides? We make them. We can break them too. So we use peroxisomes for that. Lysosomes. Lysosomes are another extremely useful organelle. The lysosome contains digestive enzymes. If you're a cell and you engulf a bacteria, if you're a white cell and you engulf bacteria, you want to fuse the vesicle that the bacteria is now in with your lysosome so it can destroy the contents with digestive enzymes. It dissolves it, it breaks it apart. We keep them, our lysosomes contain these digestive enzymes held inside their own vesicles. We don't want to release them to the cell. That would be really bad too. We would start digesting ourselves. We don't like that. We use our lysosomes to break down food particles. If a cell if a cell wears out and it's being broken apart, we'll use our own lysosomes to help break it apart and eat it. If we engulf a bacteria, we'll destroy the bacteria. If we're infected by a virus, we'll attack the virus. Sometimes we'll attack the virus and destroy it. And any toxic chemicals, we'll use our lysosomes to try and break them down. And as the organelles in our cells wear out and get replaced, the worn out ones are going to be eaten by the lysosomes. We use them also to break down glycogen to glucose and to dissolve bone to get the calcium we need. Because our body makes a distinction between strong bones and the need for calcium, and the need for calcium always wins. When a cell gets damaged, if it can't repair its own damage, it will digest itself using its own lysosomes. And we call it autolysis. Problems with our lysosomes, and there's a reason I talk about this now. The lysosomes can accumulate if we don't keep them in check with specialized enzymes. And so our lysosomes, if our lysosomes aren't functioning properly, for example, we can't keep them in check. We can't keep other structures, other molecules in check. During development of our central nervous system, we produce certain sugar-coated fats, glycolipids. We don't overproduce the glycolipids because we have lysosomes that break that down. As long as we have the right enzyme, the right lysosome to break down excess glycolipids in the brain and in the central nervous system, we're fine. However, if that glycolipid isn't removed, it builds up and starts putting pressure on the brain and pressure on the central nervous system, and ultimately usually causes death by about the age of 5. It starts somewhere in very early. It starts in infancy and usually causes death between 5 and 10 years of age. I'll leave it like that. What causes it? Well, it's not something you catch. It's something that is inherited. And the inherited trait, it's an inherited trait that is what we call a recessive trait. It's a homozygous recessive trait, meaning that the only way an individual has this condition is if they received a recessive trait from both their mother and their father. It's also population group limited. It seems to be prominent, or particularly, I don't want to say prominent. It occurs more frequently in certain population groups. If your patient, if the child that has this condition is descended through Central European Jews, don't know why Jews, but Central European Jews seem to have it occurring more frequently, much more frequently than others. So that is a possibility there. So if you have a patient that's 5 years old and having difficulty walking and speaking and has some vision problems and some hearing issues, it's something to consider, particularly if the ancestry is directly from Central Europe, with a Jewish background. Now, that's not the only, link here. Now, of course, mental, blindness, death, reduced intellectual capacity, all these are conditions. I guess death is probably the ultimate here. Somewhere between 5 and 10. Now, this condition is known as Tay-Sachs disease. Tay-Sachs disease. It is a genetic disorder primarily in descendants of Eastern European Jews, not Mediterranean Jews. And part of that was because of population behavior. Eastern European Jews were a closed society. They only married among themselves. So a large population of Jews lived in one section of a city, for example, or an area in Eastern Europe, and they only married other Jews. And what that did was inadvertently retain certain traits in the population. Recessive traits that would probably disappeared if there had been more intermarrying with other populations. Those recessive traits would have disappeared. Well, Mediterranean Jews married everybody. They intermarried a lot with other populations. There's very little cases of Tay-Sachs occurring from individuals of descendants of Mediterranean Jews because the recessive gene disappeared. Oh, it's probably still out there somewhere, but the frequency of occurrence is very, very low. Now, why am I talking about Eastern European and Mediterranean Jews here? Because another population that this showed up in is residents of some of the French-speaking provinces in Canada, particularly Nova Scotia, Newfoundland, that area. We see an occurrence of it in that population too, don't know why. But that population of Canadians didn't stay, the population that contained this gene didn't all stay in Nova Scotia. They migrated south to Louisiana back in the early 1800s, 1700s, somewhere in there. And they moved to southern Louisiana, and they refer to themselves as Cajuns now. And we've had, and if you know what recent history we know with Hurricane Katrina 20 years ago, a lot of people from Louisiana came up into this area. Now they're carrying, some of those people are carrying those genes too. So there's a greater chance of seeing Tay-Sachs disease in Tennessee and in the Southeast United States than we would expect, because we don't have a large population. You expect to see Eastern European descendants in the Northeast, but we have thanks to the migration of the French Canadians down into Louisiana 200 years ago, that gene stayed alive down there, because what did they do? They stayed in a closed population too. So there's nothing wrong with that, except it does keep recessive traits a little more viable than they would before. So anyway, so Tay-Sachs usually kills right around between 5 and 10 years of old. There are some very unusual cases that can occur later in life. If it starts later in life, it doesn't usually impact life expectancy. It does have some other similar complications here, but see, the key here is that our central nervous system is pretty well intact in its full adult size by the time we're hitting puberty. So we don't, once we get past puberty, our central nervous system isn't going to be affected by things like this, by having excess glycolipids around here. So it only has the fatal consequences in infant very early onset. So you may see this, you may not, but it's still something that we probably have a higher instance of occurrence in the Southeast US than you would expect. Okay, moving on. Also inside the cell, we have the cytoskeleton. The cytoskeleton is what gives the cell its shape. I mean, why do cells look the way they do? Why did it look columnar or cuboidal? They are held together by rods of proteins that hold the cell, give it its shape, give it its flexibility, allowing it to move. We have microfilaments, we have intermediate filaments, and we have microtubules. And the microtubules are going to form, they're going to form tubes. Our spindle fibers are microtubules. The spindle fibers we see during mitosis are microtubules. The microfilaments are the filaments that we see in our muscles. The actin protein I mentioned a couple times, that's a contractile protein, is a filament that allows us to contract our skeletal muscle. And we have these intermediate filaments that are that show up that give the cell its shape or provide certain waterproofing. This is keratin. Keratin gives us this waterproofing benefit in here by being these proteins strung together. So our skeleton, we have a skeleton in place for our cell that is totally different than bones and ligaments and tendons like we see in the adult form of us, but we still have these cells having very unique shapes. The other structure we see inside the cytoplasm are called the centrosomes and centrioles. The centrosome is the cell center. It's not the nucleus, it's the center, and found in the centrosome are a pair of centrioles. They look like barrels, I guess. And these, we have a set of them. They are at right angles to each other, and they make the spindle fibers. These are the structures that make the spindle fibers. Here at the centrosome is made of the centrioles, and they're busy making spindle fibers for us. They also make our cilia. The cilia that line our respiratory tract, line the oviduct, line our nasal cavity, and the flagella that we find on sperm cells are made of centrioles by their arrangement. Some other things that I just mentioned, cilia and flagella. Cilia and flagella are for the movement, like an egg that gets released from the ovary, once it enters the fallopian tube, has no means of propulsion of its own. It depends on cilia lining the fallopian tube to push it along. Same thing, the cilia that line our respiratory tract, for example, are going to move mucus up to our pharynx, up to our throat, so we can swallow it, to swallow away all the crud that the mucus is trapped. We also see things, structures known as villi and microvilli. These are extensions of the surface membrane of individual cells, where we want to have absorption. We want to have a great surface area. So like the simple columnar cells in our small intestine have villi, these fingers that stick up, and microvilli, tiny fingers coming off. These villi and microvilli increase the surface area of the small intestine from, the small intestine is about 20 feet long. It's about the size, about the thickness of your thumb. But if we were to lay it out flat to one cell layer thick, it would easily cover a tennis court, thanks to all the villi and microvilli. And so this is how we can absorb tremendous amounts of nutrients at one time. Now, so that's everything. We've covered the cytoplasm, we've covered the cell membrane. What we have left is the nucleus. The nucleus is the big organelle. We've all seen the nucleus in pictures of cells. It's this, even on those pictures of the cheek cells, it's the one that stained the big dark blue structure in here. The nucleus contains all of our DNA. And every cell except red blood cells has a nucleus. And even red blood cells had them during their formation, but they kicked it out to make room for more hemoglobin. Most cells have one nucleus. Skeletal muscle cells have multiple nuclei, but they don't replicate. Muscle cells don't reproduce, so having multiple nuclei is not that big a deal. What happens is when skeletal muscle cells are forming when we're in utero, they're formed through the fusion of a lot of smaller cells, and they all kept their nucleus. So we do have some cells in skeletal muscle and in the liver that have multiple nuclei. And we also see red blood cells are what we call anucleate. Now, a nucleus has three parts to it. It has the nuclear envelope, that's its membrane on the outside, same phospholipid bilayer as the cell membrane. Inside the nucleus, we have what's known as the nucleolus. That's where RNA production usually occurs. And then we have the chromatin. The chromatin is the DNA wrapped around a protein called histone. The chromatin is where the DNA is located. So the nuclear envelope, the nucleolus, and the chromatin. The nuclear envelope is, a phospholipid bilayer. It's continuous with the ER. It has openings called nuclear pores that open right into the rough ER. And the instructions from the DNA can go out the nuclear pores to the rough ER and go to work. The nucleolus is where we produce where we produce RNA. So, and you can see this here. There's the nucleolus, there is the chromatin, and there is the nuclear envelope. There's a pore right there going into the rough ER. This is what the pores look like. These little holes here on the nucleus that appear to look like holes are the nuclear pores. And then the lining of the nucleus itself looks like this. And this will be my last point because we're running out of time. Chromatin, 30% DNA, 60% histone, 10% RNA. Wrapped together, called nucleosomes, which are the DNA wrapped around the histones. It looks almost like a candy cane in its appearance. We only call the chromatin chromosomes when they condense. Other than that, it's called chromatin. So, okay. And you can see there's what I was saying. It looks sort of like candy cane with the wrapped chromatin, the DNA and the histone. Okay, we're going to stop here. And we will pick up with this. Today's Thursday. We'll pick up with this on Tuesday. Lab this afternoon. If you're in the afternoon lab, we're going to be looking at the appendicular skeleton. So, and I'm going to get us out of here, and I will see you all. I'll see most of you next week.